Thank you very much for the kind introduction. So there are two company names on this first slide, so Olympus and Evident. The reason is that the scientific solution division of Olympus left the roof of Olympus recently and now is an independent company, Evident. I'm going to show a little bit or talk a little bit about exploring high content microscopic data with the standard tool that Evident provides for this purpose. To wait to go to the next slide, sorry. Here we go. So as seen from the manufacturing perspective, high content screening means that we want to achieve highly automated microscopy in a robust and reproducible way. And typically this results in large amount of image data. For example, if we take a micro well plate as depicted here, it has, for example, 96 wells. If I want to acquire the full area or part of the area of each well, I can have easily 10 or 25 or more images per well. So I can easily end up with a few thousand images for one sample carrier. And this means two things. So we need an acquisition protocol that defines what to do and also records what has been done. So for the term of for the purpose of reproducibility, and for the term of documentation. And we need some kind of image and metadata container where all the images and all the metadata and the structure information is contained. And I will call this a scan in the next slides. Typically want to do large statistics. And typically we have a high content of information because we have microscopic images. We have maybe a 20x or 40x magnification typically. So we can have quite a degree of detail of the sample. And the challenge now is to extract the relevant information from the large set of data. And that means we need some kind of automated image analysis approach. We need something to explore the image content, to explore the whole sample. And at the end, we need also some kind of automated data analysis approach. What we call the analysis protocol in the software an assay. So I will use this term later. The foundation of any or of most analysis that we can do is the segmentation of all the instances in the images. And with instances, I mean the objects like the nuclei or the cells. And what Evident offers for this purpose is True AI, which is the deep learning based solution that we offer the users to train themselves, which can deliver highly accurate results, even in very challenging image analysis conditions. But we also deliver pre-trained neural networks for the most common applications in cell culture analysis, basically. So for nuclei, for cells, and for spots. And because that works quite nicely, I would like to show a few examples. So this is a relatively dense situation of nuclei. And we see here that the pre-trained networks finds them nicely. We see here the cytosolic staining of red 1 cells in 10x wide field imaging. Also that works quite robustly. A little bit more difficult. This is a little bit 3D situation with out of focus or defocused objects because we have one slice of a spinning disk imaging. And also here we see the nuclei detected. These are relatively patterned cells, quite difficult to analyze, but also that works okayish. And we see as another or maybe a challenging example, a very high resolution or high magnification of a confocal microscope, patterned nuclei, and something more exotic, cut through muscle fibers. See that also works with the pre-trained networks. And I think finally here an example where the cells and the nuclei are detected in the same image. I have two more examples. So this is seen from the image analysis point of view quite challenging And of course, the pre-trained networks don't deliver a perfect result here. Maybe there is no perfect result, but for a statistical purpose and analysis, this is typically a good solution. And this is finally another example of a confocal scan. So, so this was the first step, finding the objects of interest in the images, in all the many images. And now I'm going to show with a few screenshots how our standard approach is to understand and explore the sample and the data. So what we see here is the main analysis interface. What I've done before this screenshot is loading a scan, so attaching to the whole data of one well plate. And I see here in the center view one example image of the whole data set, just to give an impression what we have here. And I loaded an assay, so the analysis protocol. And the assay basically says find the nuclei. And this is what the machine did already. This takes some time, maybe half a second for camera frames. So it can take some time, but it's quite easy because no parameters are needed here at this point. So what happens then? For each nucleus, a couple of parameters are calculated, and we see here these 2D histograms and the 1D histogram here. They can be parameterized freely. In the top right, for example, I've set here for the x-axis the area, and for the y-axis the mean intensity of the fluorescence. And what we see here is a representation where each spot is at least one object or a couple of objects with the same properties. So in the lower left of the top right diagram, we see here very small, very dim nuclei. To the right, the nuclei get larger. To the top, they get brighter. And we see already some patterns. So this is probably something interesting. And to get a feeling for that, get a feeling for the distribution of properties across the sample, I can, for example, just click into the diagram, and what I immediately get then is a representative object here in the main view, surrounded by the context. So I can always see how this object looks and what the context around is the surrounding. And I could, like, if this would be like live now, I could just go through the whole data set and immediately in real time see your update as a representative in the context and get a good feeling what the data points here mean in terms of image content. I can also draw gates or regions around interesting clusters or populations of similar objects or objects that share some properties, and then get kind of a gallery view, so that I see here a random selection of representatives of the, for example, here of the top polygon. I see here the typical or random representatives, and I see here immediately on a glance what the difference here is. So this is quite bright and small. This is similar size like here, but the intensity is a little bit different. So what I see here biologically is the different states of the cell cycle. So these cells here are just undergoing mitosis or have just done so. So if I want to go further and understand more, what I can, for example, do, I can click on any object or representative that is interesting for me because it's exotic or it's typical or it's somehow unknown why it's as it is, and I click here and the whole context shifts to this object then. So I will see here the representative object in the context again. I already see it has just undergone mitosis. I see here the data in the parameter view. I see the brightness and the area. I see it's here in the top where the small nuclei are. I see it also in the other diagrams. I see here that has a average total intensity of the fluorescence dye of the G1 phase. So I see it has split already. It's consistent here, and I see also where it comes from. So with this approach, this is a very like trivial example, but I hope I can like make plausible that with this approach of interactivity and connectivity, it's quite easy to get a good feeling for the whole sample, how the properties are distributed and what the interesting and maybe rare events are. So this is already almost the end. So I've here just prepared another example, which is a little bit more complicated. So here I have another assay that looks for the cell shapes and for the nuclei. And I have done the same. I've opened here a gallery view of the cells and the nuclei, and the same idea here. I can pick any kind of object that is for some reason interesting because it has certain properties. And for example, if I randomly pick this cell here, I again see it in the context of the image. I see the properties of the nucleus of this object. I see the properties of the cell itself. And I can, like in this interactive way, explore the sample. So I'm finished now. So the next step then would be to define what are the results, but I won't talk about that now. But I think it's relatively clear that once I have defined like regions and properties, that I can easily define what is really interesting for me. All this is then saved again as an assay, as an analysis protocol, and I can apply this to the next plate and to many plates if this is required. And with this, I would like to finish. Thank you.